Humoral immunity. It sounds funny, but it's actually just really cool. Today we're going to cover the humoral immune system. This is the immune system, portion of the immune system that encompasses the B cells and the antibodies. Most people are familiar with the shape of antibodies, but don't really fully understand how they work. Antibodies are four polypeptide chains that form into the shape of a Y. The four polypeptides are two pairs of light chains and two pairs of heavy chains. The ends of the Y part of the antibody are the part that do all of the binding. This is the genetically variable region that is responsible for binding antigens. The lower portion of the Y is constant, and it actually determines the type of antibody that has been formed, and also allows it to interact with cell membranes. Now, the story of B cell production and differentiation is long and complicated, and I don't want to get into that at this point, but suffice it to understand that each B cell produces only one specific antibody, which can only bind to one specific antigen. This is the foundation of what we call the adaptive immunity system. The antibody that this specific B cell produces is inserted into the cell membrane of the B cell and acts as a B cell receptor. In order for the antibody itself to interact with the intercellular signaling system, it has to associate itself with two transmembrane proteins called IgA and IgB. This complex is called the B cell receptor and is responsible for helping to activate the B cell, something of which we'll talk a little bit more in detail soon. However, there are antibodies that do float free in the body, and these have the role of binding to antigens to prevent pathogen adhesion, and also if there's any toxins in the system, such as you might find from a enterotoxic E. coli, it can bind to the toxin itself and help neutralize it. Other portions of the immune system can also interact with antibodies, such as the complement cascade. When IgM, which is a specific type of antibody that's formed by B cells that is actually a pentad of 5 individual antibody molecules, when the IgM binds to the surface of a pathogen, it can actually activate the complement system. Now, this is the mechanism by which the classic complement activation occurs. We talked previously about the alternative complement cascade. This is the classical cascade. So what happens there is the IgM will bind to the cell surface, and in doing so creates a binding surface for complement C1. This then activates the classical complement cascade by activating C2 and then C4, which then creates the classical C3 convertase C4b C2a. This helps amplify the alternate pathway by creating another pathway for the production of C3b. Macrophages also have antibody binding regions on their cell surface such that they can bind to the constant region or the stalk region of an antibody to help it with its phagocytosis and also to help with antigen presentation. Any pathogen that is coated with antibodies offers itself up as a much more succulent treat and is much more efficiently and quickly endocytosed by the macrophages. Now, a B cell alone doesn't really have much effect as a member of the immune system. When it gets activated, it gets converted into what we call a plasma cell. How this occurs is when the B cell comes in contact with an antigen that it recognizes, and several of the membrane-associated antibodies on its surface associate with this antigen, and in addition to the presence of the IgA and the IgB molecules which serve as the transmembrane component, serve as an area of localized signal amplification that then induces a morphological change in the B cell to cause it to mature into a plasma cell. Once it converts itself into a plasma cell, it becomes much more focused on the production of large quantities of antibodies. However, as you can probably imagine, the presence of an antibody reacting to an antigen alone may be a little dangerous if the antibody mistakenly can recognize self-antigens and could cause the development of an autoimmune disease. This recognition of self is a major problem in cellular immunity, and the way in which the immune system gets around it is by using a two-signal method to act as a fail-safe. So in this respect, you think of it as cocking the gun and then firing it. Antibody binding alone is insufficient to activate and induce production of a plasma cell. In order to activate, a B cell needs to interact with either a T cell or a separate B cell specific receptor family need to be activated in order to induce maturation of the B cell into a plasma cell. So this T cell independent method requires an antibody to bind not only to the antigen, but a B cell co-receptor triad of CD81, CD19, and CR2 has to then bind to C3b in order to stimulate that B cell to activate. So as you can imagine, how this works is not only is if the B cell came in contact with something that it mistakenly thought was an antigen but was actually self, if there was C3b present on that cell as well, then it's much more likely that this is bad news because C3b is only present on pathogens. But the binding of the antigen to the antibody alone is insufficient, and the second signal is necessary in order to activate the B cell properly. The T cell dependent pathway is a much more specific way of identifying a pathogen that's invading and avoiding activating to a self-antigen. T cells are an important part of the activation of B cells and are important in general for the immune system and will be the topic of our next section. Activating a B cell with a T cell starts when the B cell comes in contact with an antigen that it recognizes on its antibody and then endocytoses the receptor antibody and the antigen. Once it's inside the phagosome, it removes the antigen from the antibody and then attaches it to another molecule called a major histocompatibility complex, or MHC. And this is a class 2 molecule. There's a class 1, and again, we'll talk about that in the next section. That then takes this MHC molecule and presents it on its cell surface with the antigen and is basically holding it up for advertising to the T cells to see if anybody else recognizes this. Meanwhile, a T cell that had recognized the antigen that had been presented by another cell type called antigen-presenting cells begins searching through the B cell population looking for a B cell that would recognize this antigen and would be able to then form an effective antibody. So to recap, a B cell presents an antigen on its cell surface after it binds to the antigen with its antibody. This binding and presenting of an antigen is on a different molecule than the antibody. This is on a class 2 MHC molecule. The B cell holds up this antigen is essentially asking the question to the rest of the environment, do you recognize this? Does anybody know what this is? Is this trouble? I don't know. Meanwhile, other cells in the area, including other types of cells called antigen-presenting cells, have also been taking up these antigens, also been taking them and putting them on a class 2 MHC molecule, and also presenting them on their surface. And somewhere along all of this, a T cell, called a T helper cell, has recognized that antigen on that surface of that antigen-presenting cell, which has caused it to become activated and go, oh shit, there's trouble now. Let's go looking to see if anybody else knows that this is trouble. Who can we alert? How can we get the word out that this is a really big problem? And this T cell starts going around looking for other cells that may have recognized this molecule, this potential antigen that could be potentially very bad news. It then comes in contact with the B cell, which is also presenting the antigen, who is asking the question, does anybody know what this is? And in effect, now you have a B cell and a T cell which bind to each other, both connecting through this antigen, both now knowing that this is trouble. This conjugate pair then move off into a different section of the lymph node and start dividing. Cytokines from the T helper cell induce the morphological changes in the B cell in order to cause it to form the plasma cell. The plasma cell is the antibody factory, and its only job is to produce large quantities and excrete these antibodies into the system. With all of these antibodies now out in the system, the antibodies bind to the surfaces of this pathogen, connecting or with this antigen on its surface, which helps serve to identify this pathogen to the rest of the body, to alert it to the source of the trouble, and allow all of the other limbs of the immune system to get to work in destroying this organism that's invaded. So today we've covered the humoral immune system. We've talked about the shape of the antibody as a Y shape with a variable region where all the action's happening up in the tips of the Y, and then the stalk of the Y, which is called the constant region, that forms that binds to the cell membrane of B cells and also determines what type of antibody it is. When B cells bind to have a antibody on its surface, this forms a complex called a B cell receptor, and this B cell receptor allows it to recognize antigens in the environment. When it recognizes an antigen, it can either use another signaling system that's on its cell surface utilizing complement cascade, or it can use a T cell to help form the cock the gun and fire it steps necessary in order for it to convert from a B cell to a plasma cell. Next time we're going to talk more in depth about T cells and the role that they play in the specific immune system.